I attended one of your talks during the Excellence Cluster Retreat, I believe last year, I don't remember, in Austria. And you introduced this kind of idea that eventually, now or in the near future, we would have microscopes that are able to recognize some pattern in your sample and then send this to another microscope that can actually start looking about this pattern, which is incredible. And here there is this artificial intelligence perhaps playing a role. Can you please elaborate on this regard?
Yeah, so I think it is something, yet another idea that comes out of the light sheet microscopy because it's so fast again. And, maybe to put it into perspective on a regular, let's say, confocal microscope, typically the process is, you've prepared your sample, you put it into the microscope, you look with your eyepiece, you find it, and then you sit down in front of the computer and you make some adjustments, and that takes some time. And especially when you're committing, when you press the start button, you have to be absolutely sure that you've done everything correctly, because you know that the next half hour or the next few hours or whatever, the experiment will be running, and there's no way for you to interfere with it. On the light sheet microscope, this is very different. The moment you put the sample into the microscope could automatically already start analyzing the sample. So basically, by the time you actually sit down on your chair, the microscope could have already analyzed the sample. It could have, for example, recognized how transparent the sample is, how many angles would need to be acquired, which angles happen to be the best, because it's not always 0, 90, 180, but it could be some odd numbers. And it could have identified maybe even, it's a zebrafish. It's 24 hours old. We looked at that yesterday. I've already learned something yesterday from that experiment. Well, now maybe we should focus more on the things that I didn't quite understand yesterday. Let's zoom in to the brain, because yesterday we tried to image the whole fish and I didn't really have time to focus onto the brain. So that ultimately is what I mean by smart microscopy, that a lot of these choices are made automatically by the microscope, based on the sample as you present it into the microscope, and you don't even have to move any sliders and find and rotate the sample. It's done automatically. But ultimately, the goal should be that, sort of as I had just said, that you want to learn from one experiment to the other, right? And that process is, you're assisted in that process by the computer, by the microscope, so to say, right? That it says, I've understood this part. I don't even have to look into that region because I've seen 137 zebrafish develop. I don't need to see this. This fish looks exactly the same as all previous fish I've seen. But over here, there's something peculiar that I haven't seen before, right? Can we zoom into this part? And the microscope would automatically record data that is something that you haven't seen before, right? And for us, sometimes it's very difficult to find these phenotypes, right? Mm-hmm. The way it's typically done is that you have a petri dish with 200 embryos and you look at them under a stereo microscope. You're trying to screen for a mutation to then, maybe find a new gene that you weren't, that wasn't known to be involved in a certain process. And even under the microscope, or in a light sheet microscope, it just becomes overwhelming because you're so distracted by all the things that you already know, and you don't really see the needle in the haystack. And in order to find that, you need some assistance. And AI, in computational intelligence can help you find that. And light sheet can produce tons of data, which is great for AI. And so ultimately, we would want to get to the point where we have a set of flamingos, a flamingo farm, a flock of flamingos, I should say, right? 10 flamingos side by side in the lab, and they just run nonstop, which again you wouldn't do if each instrument was €1 million. You wouldn't have 10 of them.
No way.
Whereas, if it's affordable, small footprint, you can easily have 10 side by side in a lab. And all you have to do is put the samples in, or that's something a robot could do. And they could run nonstop and produce the data. And they could talk to each other, right? They could be one flamingo saying, oh, seeing this in this fish over there. Do you see the same, right? And the other flamingo can look at that fish and say like, no, I mean, this fish, it looks different. And so they can have a conversation and could maybe say like, remove this sample, give me a new one. This one is boring, right? Or this one is wild type or whatever.